So in the last video we talked about gene expression, and in this video we'll talk about the regulation of gene expression. And we're going to be talking about it mostly for prokaryotic organisms, not for eukaryotic organisms. Things get a lot more complicated in eukaryotes. So we'll start with limiting our conversation to prokaryotes, right? And but understanding the process for prokaryotes does give us a good understanding of the basis of how it works for eukaryotes. Just, then there's a lot more with introns and exons, and I'll touch a little bit about how some of the process changes for eukaryotes around the end of this video. And so the genes in bacteria and in prokaryotes are kept in operons, which are groups of genes that do very similar things, right? And that is out of a concern for efficiency, right? Bacteria don't have a lot of energy to waste on inefficiencies, right? So if there's a bunch of genes that you would all want, that you want the same controls of whether or not they're expressed or not, right? Proteins that you either need all at the same time or you don't need any of them, then the bacteria put them together in an operon so that it doesn't have to have different pathways to activate all of those genes. It's to promote efficiency in the cell. And so an operon has the genes, right? So this is the lac operon, which is one of the two main examples that pretty much every textbook uses, and so I'll be using them as well. We have the lac operon, which produces genes that produce proteins that digest lactose. And so we have a operator region, which has a protein bound to it. And this protein is active at this point, right? It's doing what it's supposed to do. It's binding to the operator, right? And it binds to the operator, and then RNA polymerase, the protein, binds to this promoter. But it can't get through because the repressor is blocking it, right? So this is an active protein. And we know, right, how we can deactivate a protein, right? We can use inhibition, right? So this operon actually uses lactose itself as an inhibitor, right? And for a very good reason, right? If you only want this gene to be expressed if there's lactose. So there's two situations. If there's no lactose, then this repressor protein is active and it binds to the operator, the genes are not expressed. If lactose is present, it inhibits this repressor protein. It binds to it. The repressor protein sort of changes shape. It can't bind to the operator anymore. RNA polymerase can get through, and the genes do get expressed, right? And so then, right, and now there's a negative feedback loop, right? There's, let's switch to the pen, negative feedback, right? The negative feedback is that now the process, right, the signal, we get lactose in here, the process is it's going to copy this gene. It's going to express this gene that digests lactose. Then the proteins that are produced are going to digest the lactose, and now there's no more lactose, and we're back at this situation, right? Because there's no lactose to bind to the repressor. So it's negative feedback that, the process goes along, it gets rid of the lactose, and so then the gene stops getting expressed. Right, that's the negative feedback. Once it works, it shuts itself off. Hopefully that makes sense. So this is actually, first, before we go to that, this is a inducible operon because you need something for it to work. Right? Now, what if there's something that you want going on all the time that, say, it produces something, right? So the trip operon produces tryptophan, which is an amino acid that bacteria need to survive. And so it builds up tryptophan, and you want it to always be working, right? Because you need tryptophan, unless you have too much tryptophan, right? And so this is where the repressible operon, right? So without tryptophan, the repressor is ordinarily not active, right? Without something there, it's not active. So this is a protein that needs to be activated, right? And so this goes on, right? It produces tryptophan, and once it produces tryptophan, now we have tryptophan here. The tryptophan activates this protein, binds to the operator. Now the RNA polymerase can't get through to express the genes that are needed to produce tryptophan, right? And so again, this is a negative feedback loop, right? We don't have any tryptophan, so we start making tryptophan. But once we've made tryptophan, right, that shuts the process off. Okay, so in general, that's, yeah, I didn't think I had anything else. In general, that is how gene expression is going to work in prokaryotes, right? In eukaryotes, it's going to be a little bit more complicated. You're not going to have these operons because everything's sort of in, there's a lot more DNA. So everything's a little bit more complicated. But in general, you're still going to have the same things where you have proteins that are going to control whether or not a gene is expressed, and it will have controls. Sometimes there'll be like things like if you're producing something, if you don't have it, it goes along, but then once you have it, it activates proteins that stop the genes from being expressed. So this is just some really great mechanisms that organisms use to increase efficiency by not having genes that be produced or be transcribed, and have genes be transcribed that don't need to be transcribed, right? And so this is how organisms will regulate whether or not a gene is expressed.